Um, so just today, we're talking about plants, which sort of relates to photosynthesis. So I thought we could put them together into what I would consider a rather easy test. um, Plants are of two types. One's called a gymnosperm, and one's called an angiosperm. Um, that's okay. Maybe it's not. It's written down somewhere. A gymnosperm and an angiosperm. A gymnosperms are things that don't flower, like like pine trees and stuff. Right? They have like cones or moss, fern, things like that. Angiosperms came along a little bit later in the evolutionary process. They do flower, right? And then so that's what we're talking about today is angiosperms. And this is just like a brief introduction to plants. Um, so when you take angiosperms, you can break them up into two types: monocots and dicots. And we get this word from this, like that first leaf that comes out, we call it a cotyledon. Right. So if you look at like a seed and the first little sprout, it looks like a little leaf, but if you follow the plant as it grows, that thing's going to wilt and die. It's like something just to get some energy to the plant until it gets leaves. Um, and then monocots. So now for monocots, I think maybe grass is like a good example of monocots. If you think about the veins in the grass, they're all running parallel. Right. Whereas in like an oak leaf or something, they look like this here. Let this person in. So we're going to get to what vascular bundles are, but look at it again as like the veins. So vascular bundles are like the veins, right? So in monocots, they're all just kind of everywhere. That would be like celery, right? They're not necessarily like lined up. Um, Fibrous root system versus a taproot. So grass, if we go back to grass, it has like a fibrous root system. You can pull up grass and put it down somewhere else. Like all the roots are up at the top, right? Whereas like trees, they'll usually send a really long taproot down, and then there'll be like these lateral roots coming off of it. And then lastly, the flowers are usually they'll have like 3 petals, 6 petals, something like that. That's what monocots will have. Dicots usually they do it in multiples of 4 or 5. So that's just a comparison of the two types of plants. We often will categorize plants as being monocots or dicots, meaning that they have like one cotyledon come out or they have 2 cotyledons come out. I got nothing on this slide except that I wanted to tell you that there's 3 parts of a plant, and that's pretty easy. Root, stem, leaf. Right, so 3 parts of a plant. Let's start getting into the cellular layout of plants. So plants have 3 types of cells, and they differ in the thickness of their cell wall. So remember, plants have cell walls. So they have a phospholipid bilayer like we do, but they have cellulose around that. So the thinnest cell wall is the parenchyma. So this word up here, parenchyma. Yes, you got to learn how to spell it. Parenchyma cells, thin, flexible walls. These serve general functions. For example, the cells in a leaf could be parenchyma cells, and the cells inside of a root can also be parenchyma cells. So parenchyma cells, thin, flexible walls, general type of cell. The next one's collenchyma. So they're all ending with this word chyma. So collenchyma, you're going to find these in new growth, like a shoot, like a new shoot growth, right? Because some of the walls are thick and some of them are thin. So like unevenly thickened cell walls. That's collenchyma. So you don't find those everywhere. You just find it on where the plant is growing. Sclerenchyma is the third one, and I'm okay. I don't care about like the types of sclerenchyma. Just that sclerenchyma is the third, the third cell type. Sclerenchyma, the word sclere, SCLERE, means to like harden, like sclerosis, for example. All right, so sclerenchyma cells have thick cell walls. If we were using the box analogy, it'd be like a double-walled box. So thick cell walls. In fact, wood, what I can say about wood is that they're sclerenchyma cells. If you ever like some shells, like hard shells of seeds and nuts, stuff like that, they're made from sclerenchyma cells, so it can be pretty hard to get into it. So we got three types of cells: parenchyma, collenchyma, sclerenchyma. Plants pretty much have three types of cells. Plants have three types of tissues. So there's three types of cells, three types of tissues. A tissue is a group of cells that come together for like a common purpose. And the tissue types are pretty easy. We'll start with dermal tissue. Dermal tissue is the skin. So when you peel a carrot, you're peeling the dermal tissue. Vascular tissue is like the veins. Right, depending on where it is in the plant, they're in different places. But if we go back to the carrot, that circle in the middle of the carrot, that's vascular tissue. So vascular tissue is going to carry water and or sugars. Then whatever is not dermal, whatever is not vascular, we call ground tissue.
You call it what?
Ground. So if you look at a potato, we know we have the skin of the potato, right? And so that's going to be dermal tissue. But then you have to ask yourself, well, do you see any, like, the part of the potato we eat? Do you see any veins in there? Like once you peel the potato and it's just like white potato, or, then do you see any veins in there? No. So what is it? It's all ground tissue. So that's an example. Like potato would be ground tissue. Or on the carrot, that cylinder part in the middle, that's vascular tissue. The skin of the carrot is dermal, and then the part right underneath the skin is ground tissue. So ground tissue, I guess we're defining it by what it's not rather than what it is. It's not dermal tissue and it's not vascular tissue. It's whatever's left over. So good on that. There's a couple of types of, so let's go to the vascular tissue. There's a couple of types of vascular tissue because the plant is going to be carrying water or sugar. Usually it's moving water up from the roots and sugar is coming down from the leaves, right? The leaves make the glucose, and so they're going to travel down different organs, right? So the phloem is carrying the sugar, phloem. They're elongated cells, and they're just like a bunch of little miniature straws that are hooked up next to each other. Like this is one straw, and then this is another straw. So they're elongated cells. They're hollowed out too. So here you have a phloem cell, right? And there's nothing in it. So it's like, well, how's it living? Well, it's getting what it needs from this cell on the side, which we call a companion cell. So this companion cell is like a regular cell, and it's keeping this phloem cell alive. But you don't want to put a bunch of stuff in there. You don't want it filled with mitochondria or Golgi apparatus or nucleus. You don't want anything in it. You want it to just be like a straw that you can move like that sugar water through. So with phloem is what carries sugar. Xylem carries water. Same idea. Right, here's the cell here. Here's another cell here. So they're elongated cells, and these are dead. Like these are completely dead. All it is the cell wall. It's a hollowed out cell, and you only have the cell wall. And that's carrying water. So xylem, Xylem, carries water. Phloem carries sugar. They're usually bundled together. So wherever the xylem is, the phloem is going to be like right next to it. So they call that a vascular bundle. That was from like our first slide. They're calling them vascular bundles. They mean the xylem and the phloem together. And usually, like when you look at this, for example, the xylem is going to be internal, like on the inside, and the phloem is going to be on the outside. So it looks like there's like a little ring around here. The phloem's on like external. The xylem's internal. Here's just some pictures. So this is a root. In the root, the vascular tissue is often in the middle. So there it is. And over here, we got the same thing. So you could see dermal, right? You could see like this, it looks like one layer of round bubbles, right? It's like sometimes there's two there, but it's pretty much one layer. That's the dermal tissue. Here's the vascular tissue. And you always can tell the vascular tissue because you see those big circles. That's xylem. And then phloem's going to be like external to that. And over here, it looks a little bit different. The xylem's making like an X or cross or whatever. And the phloem is like blue. It's like in between. So all of this stuff out here, ground. It's all ground because it's not xylem. I'm sorry, it's not vascular tissue. It's not dermal tissue, so therefore it's ground. Here we'll take another look at another. These are what, stems? Okay, same idea. All these little things are vascular bundles. That one, that one, that one. Right, they're all over the place. I can see the dermal tissue. If we want to start with the dermal tissue, there's a dark, like a black line sort of around the outside. That's the dermal tissue. Then we have all these circle things, which are the vascular tissues. And if you look at one of these circle things, can't make it bigger. You see that like empty hole, like that empty circle thing there, and then there's blue on the outside of it. Same thing here. There's a circle here, that's the xylem, and you see like the little blue next to it, that's the phloem. So together they call it the vascular, right? Here it's different of a layout, but it's the same things. All the vascular bundles are going around here. So dermal tissue, easy to pick out. Now we've got these vascular bundles. So xylem, you see like these circles, like these bigger circles there? That's where the xylem is. So xylem, and then the blue is going to be phloem. And then this in the middle here, it's not vascular, it's not dermal, therefore it's ground tissue. So ground tissue on this one's all in the middle. Ground tissue on this one's just kind of, everything that's not vascular and dermal. Now let's look at some of these. Let's look at a few of these parts of the plant more closely. This is the root. All right, so they got it kind of color coded here. The blue is going to be the dermal tissue, the yellow is going to be ground, and the purple lavender is going to be the vascular tissue. So in the root, we call the vascular tissue steel. A different reason, a different word once it gets down into the root. So it's right here, steel. Because really it's more xylem than it is phloem. But they're not so much bundled together. It's just one rod. Like I refer you back to the carrot as being like a good example, right? So we call that steel. When the other day when we were looking at mitosis in a root stem in lab, what I should have done was I should have pointed you to a particular area, because roots, for example, don't just grow everywhere at the same time. There's areas where they tend to go through more mitosis. So any area that goes through like cell division, a lot of cell division, they call a meristem. So right here, meristem. So this is like a meristemic region. Meristem meaning it's going to go through a lot of cell division. Here they call it apical, which I haven't really talked about this yet, but there's two types of growth. There's apical growth and then there's lateral growth. The apex is just kind of like the end of something, right? Like this is the apex of my coffee cup, right? So if I wanted to add to the apex, I would go up that way, right? But if I wanted to make this thicker, that would be lateral. The same thing on parts like in the body and stuff like that. The apex are usually like the ends of something. So this is the apical meristem down here. That's the area that we were trying to look. And it sort of looked like this, except that we went in a little bit closer. Oh yeah, root hairs. Oh yeah, okay, so plants have a few things that they're going to do, a couple things that they're going to do to increase their water uptake. Not all plants get to be in Louisiana where there's like a ton of water in the soil, right? So they have root hairs. And if you see here, a root hair is actually part of the dermal tissue. It's just like an extension of the dermal tissue. It makes a big difference because now you can get water not just from here, but you can expand your area out. So that's one thing that a plant will do, is that plants have root hairs. The radish happens to be one that you can see pretty easily, right? You don't even necessarily need a microscope to see it. Well, my head's just pounding. Let's look at a leaf. So leaves have cuticles on them. right? That's like a waxy layer. Leaves, a lot of stems will have like this cuticle, and it's really to prevent desiccation. The plant is trying not to dry out. So that's mostly what it's for. And then we've got a few different types of cells in here. Well, you see that block of cells up at the top? That's going to be the skin, right? So dermal. Same thing here at the bottom. See that like layer of cells at the bottom? So we got those epidermal cells. And then in pink, we have the vascular tissue. So you're seeing like the xylem and the phloem. And then you got all this stuff in the middle. So there's a few things that we can say about it. One, it's ground tissue. Why is it ground tissue? Because of what it's not, right? It's not vascular, it's not dermal. Therefore, by process of elimination, it's ground tissue. Okay, what kind of cells are they? They're general cells, parenchyma. They're not like super hard cells. If I were to look at vascular tissue, that's going to be sclerenchyma. Sclere, like hard, right? The veins of something that you eat are usually like crunchy, stringy, right? So that's going to be sclerenchyma. But these are also like parenchyma cells. They're just like general cells. I was actually calling them something else in the last class when we were talking about photosynthesis. I was calling them mesophyll cells. I said mesophyll cells are types of cells that have like a ton of chloroplast in them. So that was just me making it more specific. These are parenchyma cells, but you could also find parenchyma cells like in the root, like a potato. I was saying is parenchyma cells, right? But if you look in the potato, there's obviously no chloroplast. I mean, the thing's not green, right? So how do I differentiate parenchyma in a potato versus parenchyma in the potato leaf? Right? So that's why we call it mesophyll cells. But anyway, and then there's the stoma, not to get confused with stroma, right? Stroma with an R is the liquid where we have the dark reactions. Stoma without the R is the leaf pore. So they call these things guard cells, like on both sides. And it's actually a stoma is like two cells that just it opens up like that. And then, its default is to be closed. And then it's going to do something to like open it up, which we're going to talk about in a few minutes. But those are the leaf pores. Like here on this, whatever leaf this is, you can see a lot of leaf pores, right? So they're gonna open up to let out oxygen and let in carbon dioxide. And it's kind of a tricky game because every time they do that, they also lose water, and they don't really want to lose water. But what are you gonna do? Got to get in carbon dioxide. And you got to let out oxygen. Okay. These next slides are talking about lateral growth. There, just leave it like that. Lateral growth, like woody plants, like not just trees, but whatever, rose bushes, stuff like that, right? Things that grow in thickness, right? And there's a way that they do it. So if we look at this first, in fact, if we look at this first one, this is dermal tissue out at the end. Not that we care about it. This is vascular. This is a vascular bundle. And remember, the xylem is always internal and the phloem is always external. So here's the xylem in brown. Here's the phloem in goldenrod. And then there's like this strip of tissue right here that looks gold, doesn't it? So I'll call this yellow. Xylem in yellow, xylem in brown, phloem in yellow, and then there's like this gold area, which we call the vascular cambium. This is where you have cell division. So that vascular cambium is going to divide and push on both sides. It's going to divide and it's going to push this out further, and it's going to push this in further. You can see it on the next one. So this is year 1, and then this is year 2. See that brown? That used to be here. But when the new xylem got made, this older xylem got pushed in a little bit. It's not that it pushes it in, right? Because it's going to grow out like this. Same thing on the phloem. So here's the phloem for the first year. Here it is on the second year, and there's the new phloem right here. By the way, xylem is wood, and the phloem is bark. Don't worry about the blue part right now. So this is kind of like the rings on the trees, right? There's that original brown layer of xylem. It's still working, it's still functioning, but just not as efficient as the next layer. And it's not as efficient as the next layer. So if you were to cut a tree, the wood like in the center of the trunk is not transporting stuff anywhere close to the stuff on the outside. Like that first layer of wood, like right underneath the bark, that's moving a lot of fluid. Same thing with the bark. The outer layer of bark that you were to touch on a tree, that's not moving sugar really, but the inner bark is doing that. So that's what it does. It grows from like one area, and it grows like a new layer of xylem that way and a new layer of phloem that way, and it just keeps going. We can see the rings in the trees for the xylem. Can't really see anything for the phloem. At least not us. And then some trees have bark. So I'm sorry, not bark, but like cork. So, if they do, they have same idea. They have what's called a cork cambium, and so cambium. The cambium is the area that's meristemic, meaning like the region of growth. So you don't have any questions? I don't imagine you do, because I've been kind of jumping around here.
Yeah, the only question I was having, I had in the beginning, just to clarify, you said the phloem is external and the xylem is internal.
Yeah. So just think xylem's wood and phloem's bark. So if you go back, like if I were to go back to this one, I'd be like, okay, I've got this blue thing and I've got this white thing. One of them has to be bark and one of them has to be wood. Well, that white thing looks more like it would be like the wood part, and this looks like it would have been the bark part. I don't know. I don't know. That's how I think of it in my head. but that's what wood is. Wood is secondary xylem. Well, see, there's a few things we could say about wood. It's xylem. It's secondary xylem. So they're hollowed out cells. It's just a cell wall. Your house is made from cell walls of trees, right? And what kind of cells are they? They're all called sclerenchyma cells. So your house is built from sclerenchyma cells, all the studs. Anything that's wood is essentially sclerenchyma cells that have been like compressed on each other. They're sugars. I don't know if you guys remember that, but like wood, like cell walls are polysaccharides. So, fiber. So your yeah, your house is made from carbohydrates. I mean, that's essentially what wood is. It's cellulose. I'm talking a bunch of crap now. And you're probably like, wait, is that on the test? Is that on the test? No. You have to know the three types of cells: parenchyma, collenchyma, sclerenchyma. I'll ask you about that. And you have to know, you have to be able to write something about each one. Right. So sclerenchyma is hard, like a thick cell wall. Parenchyma, thin cell wall. Collenchyma, the middle one, unevenly thick. See what I did with a question? I took somebody's question, very simple question, and I just turned it into a long thing. All right, I'm changing gears a little bit. I'm taking 3 chapters and I'm putting them into one. In fact, let me rephrase that. I'm adding just a couple of things from 2 other chapters onto this chapter. So I'm not going to go, like, we're almost done with this, as a matter of fact. So I think today we're going to be knocking off early. We're knocking off early today. Which is good for you guys. For those of you behind, like, this is your time. Like, did we do an exam last week or a quiz? We did an exam, huh?
Yeah, it was an exam.
This is another quiz. This might be your last quiz. I can't even keep track. It might not be, I don't know. But this is a chance to, again, get an A. The only thing I wanted to tell you about, I put this up here because, and I had actually done this before in the beginning of class when I was talking about atoms and water. What I was telling you was that this is called transpiration. And so let's say when one of those stomata open up to let in carbon dioxide, water goes out into the air. So from the leaf, water is going to go out into the air. And it pulls on the next water, on the next one, on the next one, all the way down to the roots and all the way down to the water around the roots. So that's one mechanism by which plants can move water through the tree or through the plant, right? Because it's got to go against gravity. You're pulling the water from the ground. How does it get up to the leaves? Because all those water molecules are attached by some kind of bond. There's like this bond holding one water to another. And it would be like no pressure, but it'd be really cool if you remembered what bond it was that attached one water molecule to another and kind of like pulled it. I know, I could just hope.
I thought it was they attached like that because water was cohesive.
It is. It's exactly it. But what are the bonds that attach one water molecule to another one to another one? Doesn't count if you're Googling it. All right, it's all right. Well, you got 3 bonds. Your choices are 3. Covalent, ionic, hydrogen. And you might be inclined to go with covalent because that would be, if you don't know the answer, go with covalent. But in this case, no, it's hydrogen bonds hold the water molecules together. So, like, I guess all these dots are they're little water molecules being connected by hydrogen bonds. And so when you pull one water, the other one follows it, and the next one follows it, and it's like a train. And we're not going to talk about the pressure. This is a stoma. There's 3 minerals that are very important for plants. There's lots of minerals that are important for plants, and they are similar to the minerals that we need. Just that they need different amounts. Like we might need a lot of zinc, but plants don't. Right? We don't necessarily need like a ton of nitrogen. Plants do. So one of the minerals is nitrogen. The other one is phosphorus. And the other one is potassium. So it's PK and phosphorus, potassium. We're going to talk about nitrogen in a minute. This is the stoma, like the opening where that lets in carbon dioxide and lets out oxygen. And you've got some organelles in here. Do you guys remember what this organelle is? The ones that are like the big sacks of water?
Is that the vacuole?
Yes. Both of you feeding my ego. Yes, vacuole. So it just stores water, right? So these guard cells have two, as you can see. And you see the cell itself is not like perfectly round. They're like elongated cells. And then, I don't know, these are like chloroplasts, whatever these are. But these right here, the vacuoles, they have got two. And so what the plant will do when you want to open up this stoma is that you want to get a lot of water into these, and then it's going to buckle right in the middle here. That's what's happening on this side. They're not showing it that much bigger, but they are bigger. Like you make these bigger, and then the middle of each cell buckles. And so how do they get water in, and how do they get out of these vacuoles? That's what they're doing all the time. They're forcing water in, and they're forcing water out, and they're using potassium. So remember, water follows solutes. We did a thing in lab where we were using salt, right? But potassium is another solute. So what the plant will do is force potassium into the vacuole. So you're making the vacuole hypertonic or hypotonic, one of those two. What's this vacuole right now?
Hyper.
Hyper. And then this area in the cell's hypo. Right, so they made the vacuole hypertonic, and that means that the water that's out here wants to go into the vacuole. And that makes this area in the middle buckle. And then when you want it to close, you just got to send the potassium out. You pump the cell pumps the potassium out, and then the water follows the potassium, and then it goes back to its original closed shape. So I was just trying to apply some other ideas that you already know to this. But yeah, these are guard cells. These are stoma or stomata for plural. And this is like a closed position, and this is open. And it's all about filling these vacuoles with more water to make it kind of bend right there. Hold on. All right. So when water comes up through the roots, there's two ways, because you want the plant to be able to move kind of quickly. The plant needs to move water kind of quickly up through the roots. So it can actually take two different routes. And so if you look at a plant is essentially one cell with like another cell next to it and another cell. They're all like touching each other. They're like bricks, right? So if you remember, I was using the analogy of cells. I say, oh, they have a balloon blown up inside of a box, right? So the balloon is like our cells. You know, our cells are like phospholipid bilayer. You could blow the balloon up too big. They could pop. They could shrivel down. Plants also have those balloons, but they have a box around it so that you don't pop the balloon, right? The balloon is blown up and it takes like form of the box. So as water, there's two ways you can go. I can go into a balloon and then go to the next balloon and go to the next balloon. And that's kind of what this is showing here. You go into the cytosol, to the next cytosol, to the next cytosol, and they call that a symplastic route. But some water hugs that little space between the balloon and the box, between the cell membrane and the cell wall. It's a little easier to move that way because there's nothing else like really stopping you. You don't have like you don't have to get in, you don't have to like bust through the plasma membrane, and you don't have to like go through the cell. We've got all these Golgi apparatus and mitochondria and all that stuff that you got to like move through. So you're just going in between the cell wall and the plasma membrane. So with the box analogy, you're going in between the balloon and the box, that little space. They call that apoplastic route. So the plant will use both methods. You can move water both ways because it's just more efficient. However, eventually the water has to enter a cell, because when you enter a cell, that's a type of protection, because the membrane, the cell membrane, is just like our membranes are, selectively permeable, meaning that we let in some things, but we don't let in other things. What if there was like a heavy metal or like a parasitic egg or something, and it's using the apoplastic route? It's going in between the balloon and the box, right? There's no way to there's no way to like prevent something bad from getting into the plant. So what the plant does, and you know, once that water gets to this xylem, once the water gets to the vascular tissue, it can go all the way up to the plant. So if there's something bad in there, you need to like find it before it gets to the vascular tissue in the root, because then it's just going to go. So they have this thing called a Casparian strip. So this is like a root hair. Water's coming in. It's not entering a cell. It's hugging that space in between the cell membrane and the cell wall. Right? So it's hugging that space. But eventually, you're going to get to like a barrier. You have no choice. You have to enter the cell. So that way, if it's something that shouldn't be in there, the Casparian strip forces water to enter a cell. Now, you ever think about, like, you're eating all these vegetables and stuff, they use like manure and stuff as fertilizer, and you're just eating the tomato. So it's like, you know, like for me, I'm not worried about it. Like, you have like a tomato plant growing in a bunch of manure. I'm still good picking the tomatoes off the top and eating the tomatoes, because I know that it's been filtered. Like nothing bad in that manure got into the plant. The Casparian strip forced water to go into the cell, and anything that, like bacteria and stuff that's in that manure, that didn't make its way into the plant. So the liquid in the tomato, you know, I would crush that and make tomato juice and drink it and not think about the liquid that was at the roots. I don't know if that's what they use. They shouldn't be using that, huh? But whatever. So plants, let me go back a few slides. Plants have root hairs, right? Root hairs allow the plant to get water further out. right? So instead of just having this area to get water, like right next to the root, it's now got this whole area where the root hairs are. But that's not enough. The plant wants to extend its reach even more. So what it's done is that it has like a mutualistically beneficial relationship. They call it symbiotic or symbiosis. There's a symbiotic relationship between plants and fungus. So this, outside these root hairs, there's fungus out here, like this, right? So the root hairs on this thing, you can't even see the root hairs, right? That's how small they are. So they're not really, they help, but they don't help as much as this fungus. This fungus is living either on or inside the plant. Either way, that fungus has itself in the plant, and it's sucking sugar from the plant. So this fungus is taking something from the plant. It's taking sugar, and it's living off the plant. But it's reaching out further and getting water and bringing the water to the plant. So they're both keeping each other alive. So yeah, this word's up here, symbiotic. It's a symbiotic relationship. About 90% of plants have this relationship. You know, when you like buy a tree or a plant or something, they tell you to take some of that soil. They don't say like take it out and shake off the soil and then plant it. They're like plant it with the soil because you want that fungus with it. Whenever they try to grow, you know, like Australia tried to do it. They got some pine saplings from the U.S. and they tried to grow pine forest in certain parts of Australia, and they didn't, because they're worried about pests and stuff. They were like irradiating it and killing off the fungus, right? And so it never grew. These trees wouldn't grow no matter what you did because they were killing off the fungus that was associated with the roots. So actually, plants have two types of these symbiotic relationships. One type is with a fungus. Here's another type. Plants also need to get nitrogen. Nitrogen is in the air all around us. You know, 78% of the air is nitrogen, but it's bonded really tight. It's got what's called a triple covalent bond. They're sharing 3 electrons. You can't break it apart. We don't. We breathe the nitrogen in and we breathe the nitrogen out. So that's what we do. All right, plants have the same problem. There's nitrogen in the air, but they can't use it. So they have a symbiotic relationship with bacteria. This bacteria is living at the roots. Just like the fungus, there's bacteria down there too. These bacteria are able to take the nitrogen and they turn them into like single covalent bonds, which are now easier for the roots to, to get from. So in short, these fixing bacteria, they fix the nitrogen. They make it into a form that's easier for the roots to take. And that's really the key behind, that's one of the keys behind us existing, because we're made of proteins, right? And where do we get proteins? And you say, well, we get proteins from like eating meat. We eat chicken. Where the chickens get it? The chickens ate worms and stuff like that. Okay, fine. Where did the worms get it? Like somebody's eating plants, right? Somewhere in there, the reason we're able to get protein, which is what we need to survive, is because of someone's eating a plant. And that plant creates some protein in it, but it can't do it without the nitrogen fixing bacteria. So it's like, the nitrogen gets moved into the plant. A cow eats a lot of grass. Is grass considered a protein? No, but grass has a tiny bit of protein in it. So like cows eating grass like all day, every day, that protein is accumulating in the muscle, and then we eat the muscle. Right? So it's really this bacteria is what allows us to do it. And I'm not even completely true with that anymore, like because now they have synthetic fertilizer, so they found a way to deliver nitrogen without just using manure. But anyway, that bacteria exists, nitrogen fixing bacteria. Um, so this is an example of like these nodules here. So these are like beans and peas, like soybeans, for example. This is probably soy. They have these like rhizobium nodules here, and they're just filled with like nitrogen fixing bacteria, right? So these bacteria are taking sugar from this root. So that's what they're taking from the plant, but they're giving the plant nitrogen, which it needs. And yeah, often farmers will grow, um, they'll grow something like corn, right? But they don't grow corn every year on the same soil. They grow soybeans, right? And then when they get all the soybeans out of it, they leave the plants and they just let them rot there, and then they till it back into the ground because you want to. That's a way of getting nitrogen into the ground. You want all those like nodules to just be mixed in with the soil. This is my last slide on this chapter. What I'm trying to show you here is that dirt is not dirt. There's different kinds of dirt. It might be hard to make out here. Can I make it? I don't think I can zoom it in. You see that it gets kind of dark up here, and then there's a layer right here, and then there's another layer down here. These layers down here are quite useless as far as growing things. So usually you have topsoil, and even above it, above the topsoil, there's often in some places like a very thin layer called humus. And that is like, you'll find that we have like a lot of leaf litter, like in woods, forest. It's just like, half an inch or something, right? And it's really rich in nutrients. It's all dead insects and feces and decayed trees, and it's just all that stuff, right? But a lot of places don't have humus, but there is a topsoil layer, right? And so that's a layer that over the years have developed a lot of nutrients in it, and that's what you can plant things in. And usually underneath the topsoil, you find a combination of different types of soils, sand or clay, or kind of a combination between the two, which is called silt. So usually we have sand, silt, or clay. Depending on where you live around here, you've got a different soil, right? Up on the North Shore, I got a place on the North Shore. It's clay. Like after I go through the topsoil, I hit like clay. Some places it's sandy. Just all depends. But it doesn't really grow anything. Like when I filled my yard, I had red clay brought in. It can't grow anything in it. It's going to take years before I get nutrients into that soil. So dirt is not just dirt. Like it depends. It's got to be the right quality of dirt. There's something you can buy in the store called loam, which is like a combination of sand, silt, and clay, and it's a topsoil. So, yeah. Okay. Do you have any questions on this? you want me to go over what questions I will ask on the quiz from this? Yes, the answer is yes.
Yes.
So let me do that. This is a giveaway question. If I ask it, 3 parts of the plant. That's just to give you a free question. And it's to weed out people that haven't studied at all. Because if you can't say root, leaf, and stem, then you deserve your grade. So 3 parts of the plant. I will ask you about this. 3, you need to know the 3 types of cells. Parenchyma, thin, flexible cell walls. Collenchyma, unevenly thick cell walls. Sclerenchyma, thick cell walls. It's like a double wall. So you've got 3 parts of the plant, 3 types of plant cells, 3 types of plant tissues. I'm probably not going to ask you anything about them because there's nothing to say. What is dermal tissue? Well, it's a skin. I mean, so just know the 3 types of plant tissue: vascular, dermal, ground. Know which one carries water, which one carries sugar. Phloem, Phloem, carries sugar. Xylem carries water. Remember, xylem cells are not even living. They're hollowed out, dead cells.
Phloem and xylem and what type of tissue is that?
Vascular. Um, know that wood is secondary xylem. Even if you put xylem, that's fine. Bark is secondary phloem. Um, what are the 3? What are the 3 minerals that are found in fertilizer? One of them is potassium. Another one we talked about. Potassium, phosphorus, and the one we talked about with the bacteria. Fine. Potassium, phosphorus, nitrogen. PKN. K for potassium. PKN. That's what fertilizer is made of. What's the purpose of the Casparian strip? If you look here, kind of where I'm circling, that is the dermal tissue. This is the water coming in. It comes in like that. Before it gets to the dermal tissue, it's got to go into a cell. That's the point of the Casparian strip. Force the water into a cell. Because the membrane of a cell acts like a filter. Last one, discuss two symbiotic relationships. One is the fungus. What's the fungus taking? What's the fungus giving? Mycorrhizae. What is mycorrhizae fungi giving? It's giving water. What's it taking? Sugar from the plant. Nitrogen fixing bacteria. What is it? What is it taking from the plant? It's taking also sugar. What's it giving? Nitrogen. It's fixing nitrogen. So you have mycorrhizae fungus, you have nitrogen fixing bacteria. Those are two examples of symbiotic relationships.
Okay, for the symbiotic relationships, the fungi take sugar but collects water for the plant, and then what would you say? What did you say was the other one that provides nitrogen?
Yeah, nitrogen fixing bacteria.
Is that just the name of the bacteria?
Yeah. Well, I mean, that's what we call it. It just fixes nitrogen. It fixes it in a way that the plant can break it up and get the nitrogen out of it.
And what does it get from the plant?
Sugar. Does anyone have any questions on today's lecture, the plants?